My name is János Percel. I'm one of the co-founders of Polygens, and I'm super thrilled to be here. Obviously, the Symposium of Rising Scholars is always an opportunity to celebrate the hard work, dedication, and creativity that all our students show in exploring the topic, going deep and understanding many challenging concepts and topics. And this is the 10th session in the STEM section of this symposium, and we are really excited to welcome our speakers, Dharmik Namineni, Indra Deshmukh, and Issa de Saint-Aignan. And we'll be getting started in just a moment. Each of the speakers will speak for about 8 to 10 minutes. Then there will be time for 1 or 2 questions. And then at the very end, we'll again open up the floor so everyone can again ask any questions that might have come up or they thought of in the interim. So with that, we'll get started. And our first speaker is Dharmik Namineni. And Dharmik, please join the stage and start presenting.
Hello. Give me 2 seconds to share my screen. Awesome. Can you see it?
Yes.
Great. So, hi, my name is Dharmik Namineni, and I'll be presenting my review paper on how climate change affects vector-borne disease transmission in Africa. So, to start with, we have to define what climate change is. And while climate change is many things, for the sake of this paper, we're going to define it as long-term shifts in weather conditions and patterns in weather events. And weather conditions and climate events are obviously really important to many regions and many parts of the world. It can especially be seen in places like Africa. And this is because Africa has an immense reliance on its environment in a lot of its societies, as well as a relatively poor adaptive capacity due to little diffusion in knowledge and technology that could help combat climate change. And one area where this can especially be seen, especially in recent decades, is in vector-borne disease. More than 80% of the population is at risk for vector-borne disease in Africa, due to its the vector's prevalence themselves, as well as the contact that humans have with vectors. First, before we go too far into this, we have to define what a vector is. A vector is simply just any way that a disease gets in contact with a host, the human host. But for us, we'll be defining vectors as blood-eating arthropods. One example of vector would be a tick, which spreads Lyme disease all through North America. Another example would be fleas, which spread cat scratch disease. And the one we'll be focusing on in this one is mosquitoes, which spread malaria and dengue. Now, the reason we're specifically focusing on malaria and dengue is because of their prevalence in Africa. According to the World Health Organization, around 627,000 deaths from malaria happened in 2020, and 96% of those were in the African region. On top of that, dengue, which is an under-recognized disease in the region, leads to severe fevers, muscle spasms, and death in a lot of vulnerable societies like young children, extremely old citizens, and pregnant women. And this is something that has to be combated since their effects will be only amplified by climate change. Mosquitoes are extremely prevalent vectors throughout the world. But in Africa itself, there's over 100 species of mosquitoes that spread various diseases. The mosquito life cycle is extremely dependent on environment, purely because from beginning to end, the environment dictates how they live their lives. For example, the mosquito life cycle early on, eggs happen to be laid in stagnant water. Mosquitoes need stagnant water to lay their eggs. Drought is something that's become increasingly common in Africa. According to the World Bank Group, it's increased 3 times in frequency and in length from the 1970s. And this is the ideal environment for mosquitoes to breed. They can lay their eggs in the stagnant water that comes from dried up rivers and bodies of water. And from there, warmer temperatures also lead to a faster development in the life cycle of mosquitoes, which means from hatching to adult and death, the entire cycle is faster. So more mosquitoes, more mosquito eggs, and a faster cycle of them hatching means a bigger vector population overall. A bigger vector population means more contact with humans overall, and more contact means increased transmission and increased transmission risk. Compounded on top of that is increased weather and temperature conditions throughout the world. The figure here shows projected mosquito population density throughout the next until 2080, and it's based on a model where climate change is peaks in 2080, and carbon emissions also peak around that time. And as you can see, Africa remains in one of the highest density and highest lengths of transmissibility throughout all 3 models. And this is because as temperatures increase, mosquitoes are able to live more easily in different environments. And increasing temperatures also drive the migration of mosquitoes, since increasing temperatures means that mosquitoes become to be uncomfortable on some environments. So move further north. Warmer winters means they can survive through these winters without dying. And overall, mosquitoes are able to survive and again transmit their disease. This can especially be seen in the transmission of things like malaria. Now, malaria is a parasitic disease whose developmental cycle is extremely based in temperature, even more so than maybe mosquitoes. And this is because they simply rely on temperature to finish the development overall, while mosquitoes can theoretically go through their entire thing and go through death, lay eggs, hatch, and go through death without high temperatures. According to the CDC, parasitic malaria simply can't complete the developmental cycle under 20 degrees Celsius. And even though projected climate changes temperature changes, are relatively little in regions throughout Africa and in other places close to the equator, where temperature is already extremely close to those limits or just above or at those boundaries, any increase in temperature means that malaria is more easily spread and malaria is more able to survive and thrive in these environments. Increased precipitation in many areas as well means that the malaria range can also increase, since humid environments are ideal for parasites to complete their development. As you can see, in just 1 year between the CDC study in 2020 and a CDC study in 2021, the malaria transmission, rate of a malaria transmission has changed. You can see in Africa, the red spot, which shows the highest amounts of malaria transmission, has increased, and whereas to take over yellow spots. And even in South America, the yellow spots, which showed only relative transmission, has now become red spots in the 2021 study. But malaria is still a disease we know a lot about. It's a disease we've been fighting. It's a disease we have surveillance. What perhaps is even more significant is the arboviruses that are starting to show themselves through Africa. Now, an arbovirus is simply any type of virus that's spread by mosquitoes. One such is dengue. Dengue is a fever, as I said before. It's a viral fever and it has a lot of severe symptoms. But the reason it's so dangerous to African society is because of how it spreads and how we look at it. Now, dengue spreads when mosquito is affected by a host. So they bite the blood of a host that already has dengue fever in it. The mosquito itself then gets affected, and then the mosquito infects an uninfected patient. This is going to be exacerbated by future urbanization that comes from increasing temperatures and increasing populations. About 60% of the African population is expected to live in urban environments by 2050. And with the increased vector population, as said before, that means that there's more hosts to infect mosquitoes and more hosts to get infected by the mosquitoes as well. On top of that, dengue happens to be a disease that's under-recognized and doesn't have much of close surveillance. In other words, people just don't understand what they see or when they get infected by dengue and simply treat it as something that they can live through. And with a viral fever, which can't be treated afterwards like malaria can, this is especially dangerous. Since preventative measures can't be taken, vaccines can't be distributed ahead of time, and important procedures to contain dengue outbreaks won't be able to be taken. And Africa as a region as a whole, of course, has many different countries and many different places, but it shares a lot of problems overall when we look at healthcare. An average of only 6.1% of the African GDP is spent on healthcare, according to the World Bank. Whereas in countries like Canada and the US, it's 12% and 19% respectively. And the reason we're using percent of GDPs is to show the value of healthcare in comparison to economies, since the cost of life and the quality of living is obviously different in different regions. And the COVID-19 pandemic especially showed a lot of the infrastructural problems when it comes to healthcare in Africa. A lot of places were simply unable to respond to pandemics and outbreaks, which led to a lot of people getting affected when they could have been prevented. And a lot of situations there have been a lot of adaptations. We've had high technology, we've had high-tech adaptations like hydrological models used to manage water flow to prevent the previously mentioned mosquitoes from laying their eggs and being able to hatch when water is controlled to make sure that there are no dry spots or no stagnant water. It means that the vector population can be put down. Now, malaria nets, another very inexpensive type of prevention, has been proven to work before with malaria, and it's shown to show extreme saturation in lower class environments, which is good because it stops transmission. Now, overall, really, what we need to do to prevent climate change's effects on disease transmission in Africa from being too prevalent is increase the focus on surveillance and education. With thousands of people dying and many people being affected, surveillance and education are the best ways to solve these problems. Inventions like the Foldscope, which are inexpensive ways to work on mobile surveillance, can help prevent and manage outbreaks overall, where education regarding hygiene and sanitization can help mitigate the impact of outbreaks while helping individual communities recognize disease and overall lessen the strain on the infrastructural system as investment goes into healthcare in Africa. And that's really it. Yeah. And thank you.
Dharmik, thank you so much for that great presentation and giving us this helpful overview. I believe we have time for just one quick question before we move on to the next speaker.
Mm-hmm.
Anyone wants to ask? Okay, so then I'll jump in. So Dharmik, how do you think climate change might affect the spread of diseases?
I think that it's going to really overall exacerbate the spread of disease. It's going to make it so that more people come in contact with more ways to get disease. And that overall will increase the amount of people that are infected and will make more and more people overall infected, and it will increase the strain on infrastructure. And it's something we have to look for and make sure to prevent through things like education and surveillance, relatively inexpensive ways to do so. And yeah, that's really it.
Thank you, Dharmik.
Yeah.
So hopefully there will be more time for questions at the very end. But with that, I'm excited to introduce our next speaker, Issa de Saint-Aignan, who will be giving the next presentation. So Issa, please share your screen and start presenting.
Hi, everybody. My name is Issa de Saint-Aignan, and my paper and presentation will be on the effects of unconscious thought on task performance. First of all, what were the main goals of the study, and what did I really want to achieve? So my first goal was to uncover what brain activity is most beneficial in decision-making and task performance. There are two main ways of thinking, conscious thought and unconscious thought. My goal was to reveal which of these two better aid performance on a task. Another one of my goals was to add to the minimal literature behind unconscious thought theory. The conscious mind is much more challenging to study as it's outside of our attention. I hope to help contribute to the research behind this less studied phenomenon. Lastly, I aim to determine the effect of time and other outside factors in decision-making and task performance. So what factors would affect the switch in thinking and which of these factors could potentially aid this performance. First, we need some background information. So what really is unconscious thought? Every thought that crosses our minds every day can be put into one of two general buckets, conscious or unconscious. While conscious thought is everything that we're aware of, or our inner dialogue, unconscious thought is everything that passes right over our heads and goes straight to our processing system. We're constantly scanning our environment, and our unconscious mind takes into account all of these things around us. This processing of information happening in the background is largely what leads to our intuition and our reflexes. Our unconscious mind also devotes time to pondering issues that are usually at the forefront of our minds. Conscious thought can largely be boiled down to all of our internal thoughts that we're not attentively aware of on an everyday basis. But how do these different types of thought come into play with decisions? When you're struggling with an important decision, people are often told to sleep on it or to hold off making that decision until morning. I'm sure everybody's heard that before. It's commonly understood that this time overnight could help to clarify the mind and lead to a better conclusion. But how can this really help? When we go to sleep, our brains actually do quite the opposite of shutting off. They go to work processing all the information from the day before, as well as deliberating on these decisions. When we're given this processing time, most often overnight, the brain's able to get a better view of all sides of the decision, leading to a much clearer view of the question at hand. While it seems obvious that there's definitely a lot of time while we sleep, that isn't the only time that the unconscious mind is at work. During our everyday lives, we complete tons of little mindless tasks that take very little attention. Whether you're folding the laundry, cooking a meal, or even just driving to work, the time that we seem to be on autopilot really gives the brain time to study everything that's happening in the background. The next issue to tackle is just finding the best way to study this idea of everything we don't think about and everything that happens in the background. The first step of diving into this idea for me was finding a study. I decided that it wouldn't be effective to run my own experiment just for the sake of time and limited resources. I decided to use an open source data set to analyze and form my own conclusions. In order to simulate the idea of making a decision, I chose to focus on either a decision or a task, and it proved much simpler to measure performance on a task just because it's much more objective. I chose a study performed by Lena Steindorf from the Heidelberg University in Germany. The graphs at the bottom analyze some statistics of just the general data set, such as sex and age. Helps get a better idea of who was involved in the study. Next comes understanding the study. So what did it really accomplish? The study I chose contained a wide variety of subjects, which led to a well-rounded data set and a little bit less source for error. There were 3 total experiments conducted in the study, but I chose to focus only on experiment 2. This gave me a narrower set to zone in on, and I had a much easier time singling out the specific factors that I wanted to study. The steps on the right outline just the general structure of the experiment. All the participants were posed with a question about a set of apartments. They would be told to make a decision regarding these apartments later. They were then split into different groups and given filler tasks depending on which group they were put in. One group was given a challenging task, one was given an undemanding task, One was given no time at all in between, and the other one was given time to consciously sit and think about the decision. All the groups then came back and they gave an answer to the original apartment task. Next came my hypothesis. I expected that the popular notion would reign true and the group given time to deliberate on the decision unconsciously would come to a better conclusion. I determined that the undemanding distraction will provide the most opportunity for unconscious thought, as little attention is required for an easy task, providing the brain with lots of time to wander and really ponder the decision. In order to find correlations and significant results in the dataset, I used RStudio and the different tools that were included. The results I found definitely surprised me, as there was not a significant correlation between the filler task and the apartment decision as I had expected. The participants who performed an undemanding filler task did not show better decision-making. There was, however, a significant correlation between the type of filler task and the performance on that filler task. Those who were given a more difficult task commonly performed much better on their task. This did seem counterintuitive, as a harder task should yield lower scores. But upon analyzing this information, I determined that the more difficult task really kept the participant's brain engaged and in active thought. While many of the thoughts recorded were totally unrelated to the task, they overall had more thoughts than those in the undemanding group. The undemanding task seemed almost too easy and let the participants' brains kind of check out, so they weren't recording as much thought at all. I believe that the demanding task was just the right difficulty to keep the brain active while not requiring enough brain power to shut down outside thought entirely. This helps to prove the correlation between unconscious thought and performance, as those who were engaged by the task, whose brains were stimulated, and who recorded more thoughts, whether they were related or unrelated, performed much better. When the brain is active, even while wandering, much more success with the task was recorded. While these findings scratch the surface of what our brains are capable of, the research doesn't stop here. One more trial that could expand on this experiment could introduce a third filler task that's even more difficult than the second. This could test the limits of what difficulty makes unconscious thought impossible, and potentially find the area of thinking that fosters the most unconscious thought. The most difficult aspect of this field is finding a way to effectively measure something that's not obviously tangible. Bridging this gap on how to study the phenomenon of unconscious thought effectively will open the door to groundbreaking understandings and revelations about the workings of our minds. Concluding, this study only illuminates just how many of our everyday actions are so far out of our control. Research has shown that some 95% of all thoughts are unconscious. This study outlined how these unconscious thoughts actually work towards our advantage. When we're posed with a difficult decision, task, or situation, the brain goes to work in the background to make sure we fully grasp what's happening and see all sides of the problem. This can help us form a better conclusion or perform better, all just due to unconscious deliberation. Research will only continue from here to provide an even better idea of the inner workings of our brains and how we can harness that to truly know ourselves, inside and out. These are the sources that I use for my project. Thank you guys so much for listening.
We have up next Pratham Chhabra. Pratham, are you ready to share your screen?
Yep, thank you.
Great.
All right. 4 million athletes experience concussions annually. In fact, according to a study from the University of Pittsburgh, more than 300,000 of these traumatic brain injuries stem from American football. Inherently a violent sport, football is known for its constant savage tackles and brutal helmet-to-helmet collisions. But how can we stop this and the accompanied brain degeneration or chronic traumatic encephalopathy that follows? Hi, I'm Pratham Chhabra, and I'm here to solve that problem. Today, I'll be walking you through what exactly is chronic traumatic encephalopathy, or CTE, the impact of it on the sport, the problems with concussions in football, and a comprehensive 3-step method to solve this critical issue. Next, I will look to discuss my research through my findings and through the insights I extracted from my analysis process. And finally, I will be honored to answer any of your questions. But before we begin, I would like to give just a quick background of myself. Being a passionate sports fan since I was just 5 years old, I hated seeing my favorite football players fall onto the field after injuring their head. And that's why I wanted to embark on my study to solve this problem. CTE, a form of brain degeneration, is caused by repeated brain traumas or head injury, such as concussions. Despite the use of helmets, due to repeated impact, CTE is quite prevalent among the sport's most idolized stars. In fact, the United States National Library of Medicine explains that CTE causes a progressive decline in memory and cognition and contributes to depression, suicidal behavior, and puts someone at eventual risk for other diseases such as dementia and Parkinson's. And why can't brain scans be done before death, is what you may be asking. But there are scans that exist to determine brain volume loss on live patients, not those who have passed away. So full confirmation of CTE cannot be done until the brain examination after autopsy. Now, as many of you may be familiar with, concussions are the immediate impact of brain traumas on the head, which are repeated blows that do cause this brain degeneration. And the immediate impact of such injuries does not go unnoticed. Although unlike a progressive decline in the protein of the brain, headaches, confusion, lack of coordination, memory loss, nausea, dizziness, and excessive fatigue are all potential symptoms because of concussions. With helmet-to-helmet contact occurring with such an astronomical tendency, numerous concussions actually go undetected or are not properly addressed by medical officials, threatening to induce CTE later in these athletes' lives. In fact, head injury experts explain that 95 G-forces, one G-force which represents the standard gravitational force on Earth, are enough to suffice for a concussion. And believe it or not, the average football tackle or hit exerts 105 G-forces of strength. So you can see that these hits actually outweigh the amount of G-forces that take to cause a concussion. And clearly, there's a problem. Simply take a look at the emotional and physical toll the sports take on its players. Look at Hakeem Johnson here as he falls to the ground in a Super Bowl match after he loses all consciousness with such a brain injury. Many of the complications associated with concussions and progressive brain injury are preventable. But currently, what even are the problems? Well, countless of these injuries are a result of inefficacious helmet technology. None are engineered for concussion elimination. Secondly, safety is simply not a concern. Ultimately, many of these injuries stem from dirty or unsolicited hits, inadequate training, or even ineffective gear. Per health organizations worldwide, these injuries can be prevented. However, it is evident that proper equipment, guidelines, and training must be integrated into the game in order to do so. Now, in order to analyze chronic traumatic encephalopathy risk in American football, without actually viewing brain scans, I contacted the National Football League's very own Health and Safety Department and was given access to their database of records. Spanning from 2012 to 2015, we used a 3-year sample of data to conduct our research. As such, we used Python's data filtration capabilities to parse through the 10,000 individual injury records. First, we used the Python's Panda data frames to package or refine our statistics to strictly concussions, which again are a form of brain trauma. And this allowed for maximum alignment to the brain degeneration insights that we saw. Subsequently, we retrieved each athlete's name, position on the field, association to a team, total games missed, and finally, what season the report is taken from. In doing so, this numerical data manipulation process gave us the appropriate setup to conduct an organized data sheet. Through separating each year of records into 16 distinct columns, the appropriate assortment of figures was achieved. With the respective year, total concussions, groups listed, the organization allowed for the most efficient process. Well, what did I concur through my research? First, I made crucial chronic traumatic encephalopathy predictions in various football positional groups. Predictions on the wins above replacements, or WAR metric, to determine the impact of unhealthy players on a team. And finally, a helmet to prevent concussions that I actually rendered or designed myself. Through my data analysis process, as can be seen through the various graphics, I uncovered crucial data. Quarterbacks, or the players who throw the ball, are showing 96% more signs of affecting team win percentage significantly than other positional groups. On the other hand, positions like cornerback and safety, which play defense, account for 29.4% of brain traumas. And my biomechanical simulation demonstrates that each head-to-head hit produces 105 g-forces of magnitude, where concussion's threshold is just 95 g-forces. In fact, per the research, 99.05% of National Football League athletes are at risk for chronic traumatic encephalopathy and brain degenerative dementia, signaling such cognitive dysfunction. With the following positional groups most susceptible to concussions, it can truly be seen that the game of football is not a good place because it is affecting fans and the game at hand. But unless we put a stop at it now, the problem can be solved. Introducing football's first helmet to eliminate concussions. Through working with the sports analytics majoring student at the University of Syracuse to discuss the essential components of a helmet designed for durability in American football, I researched chronic traumatic encephalopathy design technology that can be implemented into headgear. Next, I compared my industry knowledge to current helmets and used high-grade materials as a final engineered structure, as can be seen in the render. The components of the helmet are developed using the hardest shells of polycarbonate possible, as can be seen, accompanied by ABS thermoplastic polymer, a G-force sensor, Rheon intelligent energy control cells, and precision fit lining layer. The helmet prioritizes safety. In fact, the G-force sensor extracts real-time insights to analyze the magnitude of the force to a helmet. Working with the energy control cells, they compress or shear to absorb energy at the impact site. Through using university-level biomechanical simulations of the helmet in action to test impact absorption, I note that the helmet is safe, effective, and most importantly, revolutionary. In conclusion, brain degeneration is an inherent part of the game and continues to be because of the undiagnosed concussions among the sport's biggest stars. Through implementing a helmet that eliminates concussion risk, however, it can be seen that team success is greatly altered. If key athletes are missing, teams have a lower wins above replacement metric because of the magnitude of this issue. But with important rules, player training, guidelines, and helmet, the league is sure to thrive. And now I would be honored to take any of your questions.
Thank you, Pratham, so much for that presentation and the exciting and highly relevant work you've done here. Any questions from the audience or the panelists? So there's a question from the audience, which is asking, if we want to prioritize safety, then why not shift to why not change the rules or go with another version of football?
Yeah, so that's actually a great question. So before I actually started this study, that's exactly what I was thinking of. Why can't the National Football League and other leagues implement rules to stop the spread of concussions? And actually, they have taken measures to do that, which include penalties such as unnecessary roughness or tackles after the play is over. But however, the problem is that these guidelines only ensure that plays are not dirty or don't have unsolicited hits, and rather these tackles still occur and affect the brains of these players. So the only way we can truly stop that is with helmets, or simply put an end to American football, which as a fan myself, I would definitely not want to see. So that's why I constructed that helmet to make sure that these injuries potentially do not happen no longer.
Pratham, super exciting and really excited to see where this helmet takes us in the future. Hopefully there will be more questions towards the end that we can bring to you. With that, Pratham, thank you so much for the presentation and for doing this great work.
Yes, thank you for the opportunity.
Thank you. And up next, we have Hassanatu Kamara. Hassanatu, please join the stage and start presenting. All right, feel free to start.
Sorry. Can you guys see my screen though?
Yes, we can.
Hi, my name is Hassanatu Kamara, and I'll be talking about CRISPR therapy for gastric cancer. I choose gastric cancer because it is responsible for a high amount of death globally every year. In this presentation, I'll talk about its current treatment and the therapy that might be the best option. Gastric cancer is a common disease with high mortality. It can affect any part of the stomach. This disease was first discovered in the 90s, and it is a disease that causes an unusual extension of tumors in the stomach. Some of the signs include poor appetite, weight loss, heartburn, vomiting with or without blood. According to gastric cancer death estimate in 2020, as you can see in the table above, 768,793 people died in 2020. That's a large amount of number. But however, we'll be discussing about numerous risk factors in the following slides. But before we go further, let's talk about cancer in general. Cancer is the accumulation of DNA mutations, the overgrowth of cells that become a tumor, and the eventual metastasis which affect multiple organs. There are numerous environmental and hereditary risk factors. Environmental risk factors include obesity, high salty and smoked foods, infections with Helicobacter pylori, and smoking. And the hereditary risk factors include hereditary diffuse gastric cancer, CDH1 mutations and mutations in other genes such as BRCA1 or BRCA2, and Lynch syndrome, which refers to like family restoring genes of cancer. Surgery and chemotherapy are first in line treatments for gastric cancer. But however, surgery is suitable for all types of cancer or for all situations. It isn't feasible when the tumor has spread. And for chemotherapy, chemotherapy is good for advanced tumor cancer, and it can relieve cancer symptoms. Chemotherapy can treat cancer by inserting medication like pills or liquid into the bloodstream using a catheter. This treatment is usually given every 2 or 3 weeks, depending on what drugs you're taking. You have to go through chemotherapy to feel relief for a certain amount of time once you started taking this treatment. But however, it can't cure the disease gastric cancer. While these are useful for some patients, it doesn't work for everyone, and we need more options. However, there's this new technology that can be used to edit and repair DNAs. And this technology is called CRISPR. Now let's talk about it. I know you guys might be wondering what CRISPR is. Well, CRISPR is the technology that can be used to edit and repair DNAs. CRISPR makes it possible for us to move genes from any living thing into another one, altering DNAs for generations to come. It can be used to knock out genes, especially oncogenes, and prevent cancer cell growth. It can be used to repair mutated genes, tumor suppressors like p53, and promote cell death. It is a way of finding specific bits of DNA inside a cell and it is often preferred because it is inexpensive compared to other methods. This technology is composed of two main parts: the Cas9 protein and the single guided RNA. The Cas9 protein is a non-specific nucleus protein that cuts DNAs at random places. The other component is the single guide RNA, which is a single-stranded RNA molecule that targets Cas9 proteins to the correct DNA sequence by sequence complementarity. The term refers to a sequence of bases that will usually form a double-stranded structure by identifying the pairs. However, as you can see, there is a figure at the side of the slides that shows how CRISPR cuts and repairs. We have a double-stranded DNA in this figure, and we have a Cas9 protein. But the Cas9 protein is unable to recognize a specific DNA target. But when the single guide RNA is present, it will direct the Cas9 enzyme to the region, and the cuts happen from there. To get CRISPR into a cell, we need a system of delivery. One of the system of delivery is called viral delivery. Well, viral delivery has transduction efficiency. It has transgene expression that can be controlled by virus, and it can target specific cells types, such as dividing cells or non-dividing cells. Viral gene delivery system consists of viruses that are modified to be replication deficient, which were made unavailable to replicate by redesigning, and it can deliver genes to specific cells which can produce expressions. These viruses can be injected or given by IV directly into specific tissue in the body, while it's taken up by individual cells. The cause of viral delivery includes immune reactivity. Depending on location of tumor, delivery can be harder. And cytotoxicity, replication competence, no targeting, and small insert size is also some disadvantages of viral delivery. Nanoparticle delivery can also be used to deliver CRISPR and nucleic acids. This is a new technology that was recently used in COVID vaccines. This was delivering the mRNA as opposed to the DNA. Cause of nanoparticle delivery includes bioavailability in advance by increasing aqueous solubility, drugs are targeted to specific locations in the body where there should be a delivery, and the disadvantages include inflammation in the lungs and heart conditions. Nanoparticles used to carry drugs to the brain may be toxic. The future of CRISPR in cancer and its limitation. In cancer biology, CRISPR technology has a bright future ahead of it because the technology is adaptable, it is simple, it's convenient, and it's efficient. However, the downside of CRISPR include off-target effects. Off-target effects are major effects of CRISPR. And CRISPR can be used to treat other diseases. It could be used to treat diseases like sickle cell disease. At the side of this slide, you can see a figure that shows how CRISPR can be used to repair DNAs in order to cure sickle cell anemia. CRISPR can be used to cure sickle cell by taking stem cells from a patient's own body, treating 40 genes with them, and then repairing the other stem cells into the body to alleviate symptoms and genetic disease. This method introduced a novel approach to cancer treatment by allowing for modifications to genome of target cells, which was previously difficult to achieve. Scientists and experts have predicted that this technology will transform our planet and our societies. Thank you. A special thanks to my parents, Mr. Garrett Cooper, my mentor, and also Polygens. These are my sources.
Hassanatu, thank you so much for that presentation. And you know, as someone who has had many people with cancer in the family. All these steps forward with CRISPR and other technologies is much welcome. Questions for Hassanatu? Quick question from me then. Are these technologies already being used for cancer treatment, or these are just more on the horizon?
CRISPR is on the horizon. Although some people have been using it, but they are still conducting research on it to get to know correct some of its flaws so that it can be a technology that people can actually use and depend on.
Great. Great. So I'd like to ask all the panelists to come back and start streaming their videos so we can start asking them questions from the audience, from each other. Yeah, I'm really excited by all the incredible presentations that we had here today. Obviously, these represent tremendous work by all the panelists over the past few months. We celebrate that. And we will now open up the floor for questions. So Lily is actually asking all of you what the most challenging part of your research was. So let's go in in the order that the presentations took place. So I believe then Dharmik would be first, right?
Probably one of the hardest parts of my entire presentation, or my entire paper really, was figuring out exactly what I wanted to research and what specific parts of each section I wanted to research. There was a lot of information on the field out there, but a lot of it was very general, not very specific and not very numerical that I could actually analyze and write into a paper. So delving into sources and looking for the proper ones to use. It was a really hard part, but my mentor really helped me through it, and it was a good time for me. I learned a lot.
That's great. Isa, how about you?
I think one of the most challenging parts of my paper and the whole process was probably kind of analyzing the data myself, because I used an open source data set, and obviously the scientists who had collected that data and analyzed it on her own had found her own findings, and there was a lot of trial and error of using the data and finding correlations like, oh, I think this might be significant, and then realizing that there really wasn't any correlation between those things at all. So it was definitely a lot of trying out different things, thinking about what could be correlated, and then kind of analyzing it on my own. So yeah, definitely finding what really mattered in that data set and what was important.
Makes sense. Indra. Just to repeat the question, what was for you the most challenging part of the project?
Oh, sorry, I wasn't sure whether my microphone was on. I think the most challenging part of the project was probably going out and collecting the data myself, because as I was going, I realized that a lot of things that I had, like patterns that I had expected to see, didn't exactly match what I was actually seeing in the environment. And of course, going out and looking at an ecosystem, you're never going to find something that perfectly matches a scientific hypothesis. So I think, reworking my ideas based on that was pretty challenging for me.
Makes sense. Thanks for sharing. Pratham.
Yeah, so for me, it was actually figuring out what are the constituents of a helmet that can actually prevent concussions. So as you saw, the helmet that I designed had a ton of different parts that seemed to all be moving around. But the fact of the matter is the current helmets on the market, they're all so different and unique in their way, whether that's structural or the technology. So actually it was kind of challenging for me to research what materials should I use, what shape I should have the helmet as, and how should I use the statistics on concussions, and even the study of the brain itself to figure out what helmet would be most beneficial for the players.
Makes sense. these are all great challenges with these research projects. Hassanatu, how about you?
Well, for me, it was like to organize the document and like to make it make sense, because you know, cancer like it's a broad topic, and it like we have like a lot of different type of cancers, and trying to like figure out like what I should use and what would be best for the research, that was kind of challenging to me. And also I have to find out gene that cuts and repairs, because like I'm talking about CRISPR. I have to like make figures that will match with what I'm saying. So that was all kind of hard because it's all new to me.
Makes sense. Well, thank you all for sharing that. So I think we have time for another round of questions, and we have a question from Amber. Actually, I think I can let some of our attendees ask the question themselves. So Amber, if you're here, you are now, you can ask the question.
Hi, everyone. I hope you can hear me. Okay.
Yes.
Great job today. My question is, what was most surprising during your research process? It can be a surprise finding or surprise in something learning about yourself. So you can take that question whatever direction you'd like.
Thank you for that question, Amber. Let's go in reverse order. So let's now this time start with Hassanatu.
So I was surprised by all this CRISPR technology thingy, because you know the famous treatments for cancers are mostly chemotherapy, surgeries, and immunotherapy and radiation. So getting to know about like CRISPR and how it can like magically manipulate DNAs, that was all surprising to me, because like I never thought there was a technology like that.
Got it. Great. So next one is Pratham.
Yeah, so first of all, I was surprised about like my resiliency, because research can always be like a tedious process. So you have to continue to work. And I'm sure all of you like share that same experience with me. So that was definitely surprising for me, like on a personal level. But when it comes to the research itself, what I was most like surprised with was the data where 99.05% of all American football athletes are actually at a risk of C.T.E or the brain degeneration.
Wow, the statistics it kind of justifies spending some time on trying to come up with better helmets, right?
Yes, indeed.
Got it. Indra.
One thing that really surprised me was just the amount of effort that goes into removing an invasive species, because in my area, there is a volunteer team that's been going around clearing the species every single week for several years. And it's just so much work is required, even going back to the same areas every once in a while and redoing them. And it really shows that once an invasive species gets established, it's so difficult to remove it that it's better to just prevent them from being established in the first place.
Great learnings. Isa.
Yeah, I think what was most surprising for me is that I actually did have kind of like a shift in my research. I had one hypothesis that I thought that one certain section of the data, I thought that one different condition was going to yield the best results. And then once I went into the data and started analyzing that, I realized that my hypothesis was actually not true, and that didn't really have any significance at all. And when I figured out what was significant, the pieces kind of came together of how it is basically a reworked idea of my hypothesis. So it displays like the same ideas, but it just came out in a different way. So once I kind of pieced together why the results that were significant, there was kind of that moment where you're like, that makes sense. And you can see how the principles kind of apply just in a different way, which was really cool to see play out.
I love that story that highlights that light bulb moment that you have as a researcher trying to make sense of all the results and the mass. Great. Dharmik.
For me, it was how much I enjoyed public health and learning about public health. Before this project, I hadn't really put much thought into the field. I was still kind of deciding what I wanted my major to be in college. And learning and going through the entire process of researching and learning about how like communities interact with diseases and how they interact with the biology around them became something that's super interesting to me. And now it's something I'd love to major in college. So I think I learned a lot by myself in that way. So that was cool.
That's a great personal reflection, and love hearing that. I think that's the best outcome. You spend a lot of time on something and then you decide to take it to the next level. Great. So thank you all for answering those. We are way over time, but any further questions from the audience or panelists to each other?
I had a quick question for Indra, if that's okay. Because I used a data set that already existed. I was wondering how you did like some live data, and I was wondering if that was how that was for you, like going into the field, and if you think it like expanded your idea on it, because I think that would be something that's really interesting to do.
Yeah, I think it definitely did. I based everything off some existing research that I looked at beforehand, and then I developed like what I was going to look at based on that. And yeah, it was really interesting going into the field and then seeing that things didn't exactly line up in real life, how, they would look on paper. There's never going to be like a 100%, equal species diversity suddenly showing up anywhere. And I also had a lot of help from the Walnut Creek watershed, who guided me through the different sites that they'd been clearing. So it was thanks to them that I was really able to get that done.
Yeah, that's incredible that you went out there. As I noted earlier, I think doing field work as a researcher is a real test of determination. Great question, Isa. Any other questions? Question to Pratham. So do you plan to build this helmet?
Yeah, so actually I was thinking about that, to build a helmet or to not. So considering how expensive all these materials are, I think to potentially like build one as a prototype, because although I don't play football, a lot of my friends do for my local high school. So I would definitely like have them try it on and try using it. And then potentially, since I would want to pursue a career at the National Football League in the health and safety department, potentially having them implement my ideas into the game would be something that would be maybe like a dream for me there.
That's great. Long-term goals there.
Yeah.
Fantastic. Great. So any last minute questions from the audience or from the panelists to each other?
I just have a question for Hassanatu. So I was just wondering, like, what inspired you to kind of like start your research on CRISPR and like biology in general?
Yeah, so I want to pursue a career in the science field. So first, when I wanted to like start a research, I was kind of confused on what research to do. And since like I've lost like a family member to like gastric cancer, I thought like that would be like a good idea to just know like some basics about this type of cancer, how it works, and possible treatments that can help to cure like the disease in the future. And then I got to know like a little bit more about CRISPR. I expanded like more than I thought, because I just wanted to know a little. But like with this research, I just, yeah.
Got it. Any other questions? Dharmik, what made you focus on vector-borne diseases and climate change and the climates?
So climate change has been a really big interest of mine since I was really little. Just the environment and how it's been affected by everything that's been changing in the world. I've lived through a lot of changes too. I thought it's just always a really interesting topic, and going into it, I just came out of AP Bio, and biology also was something that interested me, especially the microbiology of it all. So my mentor gave me a couple articles, and that really helped me narrow it down to I want to do vectors. And I was originally going to do ticks until I read an article about how bad malaria outbreaks and dengue outbreaks were getting in Africa. So I decided to do a little more research into that and write a paper on it.
Got it. Well, a lot of issues on our minds that can spur those new directions we can explore. Any more questions? If not, I have a final question for Isa. So has your research changed how you make decisions and think of your unconscious functioning?
It's definitely made me more aware. I think one thing it changed a lot too is that, especially in high school, people think like, oh, I have a test tomorrow, I need to stay up all night studying for this. And it also kind of points out that like cramming for things like that doesn't really help, because your mind needs the time to rest where it can process that information. So almost just as important as the time you spend studying for something is the time that you give your mind while you're resting or while you're doing something that doesn't take as much thought to kind of process that information. So yeah, I think it's definitely changed the way that I think about things, and it's brought a couple more things to my attention that I wasn't really aware of before.
Well, I think that's a great final note. I think we all need to process these great presentations, the work you've done, the incredible answers you gave to questions. I think our subconscious is right now overloaded with new stimuli and the information. So it's time for us to take things offline and process all that. Again, I wanted to thank all the presenters today for doing such an incredible job, not just today, but over the past few months, and bringing all these learnings to us. This is the moment when we celebrate and cheer you. Thank you so much. And with that, we'll conclude this session of the 7th Symposium of Rising Scholars. And of course, feel free to reach out to the speakers after this conference ends. You can email anyone on the Polygens team, and we'll be happy to coordinate introductions. Thank you again. Have a great day, and onwards with more research, exploration, and discovery, which I think will just make this world a whole lot better place to live in. Thank you for joining us today. Bye.
Thank you.
Thank you so much.
Thank you.